Okay, let me share my screen to all of you. Okay, so in last class we solved the question up to this. So if the slide is visible to all of you, then please do write in the chat section whether you can see the question or not. Then we will start with our session. Can you all see the slides? Okay, so let us start. This is a question from week 2. We solved till question number 22. So this is your question 23. So this is a very important question. So that's why I wanted to discuss this in today's class as well. So it states one side of a 30 residue alpha helix faces the hydrophobic interior of a protein. If the helical wheel of this helix were to be drawn, which of the following residues are likely to be hydrophobic? So these are the residues, these are your options. So basically it is stating that the alpha helix consists of 30 residues of amino acids. So you just need to draw a helical wheel for 30 residues and then guess that what is the probable sections or probable residues that are likely to be stay in the hydrophobic region. This is a very interesting question. So for all of you, first I will try to draw the helical wheel because this kind of question will be very important for your exams and in general understanding as well. So first try to draw the helical wheel for 30 residues. Just number it like 1, 2, 3, 4 like that. You do not need to exactly know the residues, okay? Just draw the wheel for 30 like from 1 to 30. What will be the helical wheel? And then try to find out the section which should be hydrophobic. I will discuss the question thoroughly. Just try to draw the helical wheel. Okay. It will not take much time because I think up to 15 residues it is already shown in your lecture material. So you already have that. After that, after 15, just put that. So helical wheel is basically very easy. 1 second. So if you have your circle, that is a very bad circle, sorry for that. So you have to just divide it like that. Let me draw it again. I drew a very bad circle with mouse. So you have to just divide it at different angles. So if it's 0 degree, it is 90, it is 180, sorry, 0, 90, 180, 270 and 360 like that. So in helical wheel, what we do? If we place our number 1 residue here, then the next residue will be at an angle of 100 degrees. So if it is the 90 degree angle, so suppose it is the 90 degree, then it is your 100 degree. So the next residue will be placed here. And so on, the helical wheel continues. Again after 100 degree, you have to place your next one. The basic thing for this question is to see whether you can draw the helical wheel or not. This was the basic purpose for this particular question. If you can draw that correctly, it will be very easy for you to identify the correct option. Okay, so if you have done that, I will show you. You can match your answers with mine. So the answer should be D. I will come back to it. Again, sorry for the bad handwriting that is written here. So basically, if you see, I have already drawn the helical wheel here. So this is your whole circle, and suppose it is 0 degree, and from that the whole angles are written all over here as well. So first one we have placed at 0 degree, that is number 1 residue. The second one will be placed at 100 degrees, that I have placed here, number 2 residue. Then next angle will be 100 degree more, that is basically 200. So we have moved to 200. This is your residue number 3. Again 100 degree, so that will be 300. You have your residue number 4. Again you have your next one, that is 400. So up to that, this is your 400. So here you can place your residue number 5, and again at angle 500, and so on. You have to just add 100, 100, 100 and place the respective residues. So once you have done all the placing all the residues from 1 to 30, I have placed all of them. Now if you look at the option that was given here, these are your all the options. So see, one thing Professor Dasgupta has mentioned. So suppose this is your helical wheel. Okay, fine. So either any of the sides of this helical wheel will be hydrophobic, or any of the sides will be hydrophilic. Any of the sides, you know. This side can be, this first half can be hydrophobic, this lower half can be hydrophobic, anything can be hydrophobic. Because you do not know the sequence in this particular case, like I just give you the numbers like 1 to 30, but I did not give you the sequence, so it is not possible for you to predict from the sequence that whether the residues are hydrophobic or hydrophilic. You just have the numbers 1 to 30. But one thing you know, whichever side is hydrophobic or hydrophilic, that numbers will stick to a certain part of the circle. If this particular part is hydrophobic, then all this number will stay together as a hydrophobic residual part. And if this part is hydrophobic, then these numbers will stick together in the option as a hydrophobic part. Now if we look at our options, see, our first option is 1, 6, 11, 16, 21, 26. So let us go back to the wheel. It is 1, then another one is 6, then another one is 11, another one is 16, another one is like 26. So if you follow that, you can see that the residues that are mentioned in the option are all over the circle, just randomly placed. So it is like the hydrophobic units are everywhere. But that is not the case for helical wheels. Helical wheel is designed like that, so you can divide the helix in parts. But if you go through that option, you can see it is randomly scattered in your wheel. So the first option is obviously not correct, because the entire part randomly cannot be hydrophobic for helixes from helical wheel predictions. Okay. So our first option is not matching with the concept that we already have in case of helical wheels. Let us look at the next option. It is 1, 4, 7, 10, 13, 16, 19 and many more. Let us look at the first one, 1, 4, 7, 10, 13, 16. So this is our wheel. Again 1 comes here, 4 is here, here is 7, 10 and 13. Again it is taking the entire circle as an option, randomly scattered option. So again the second option will not be correct, because basically it is taking up all the random portions of the helical wheels circle. Okay. So again that can't be our option. Let us move to the third option. It is 10, 20, 30. Let's go back to it. 10, 20 and 30. So here is your 10. Let us look for 20. Here is your 20 and here is your 30. Again, it is like a, if 10, 20, 30, if we take this region, if they are saying this region should be hydrophobic, why it is not true? Because if 10, 20, 30, if gives us this right side region of the circle, then all other residues of this helical wheel should have been in the hydrophobic part as well. Because basically you are just dividing a circle in two halves, and one half will be hydrophobic and other will be hydrophilic. So the option says 10, 20 and 30. So they are basically dividing it here from the circle. But they have not included the other residues that are already sitting in that half, like 17, 6, 24, 13, 20, 2, 9. They have not written those. So again, that option is incorrect because it has not included all the residues that were present in that section. Let us look at the last option. It is 1, 5, 8, 12. Let me write it down so it will be easy for me. 1, 5, 8, 12, 15, 19, 23, 26, 30. Let us see if they stick together in a circle or not. So first is 1, fine. Next is 5. Let me 1 second. Pen. Just choose a different option. Sorry for that pen. Okay. Let me use the highlighter in this case. So first is 1. Okay, we have got our 1. Next is 5. So this is our 5. Next is 8. Let's look at it. Here is 8. Fine. Next is 12. So this is our 12. Next is 15. So if we look, this is our 15. Next is 19. So again, this is 19. Next is our 23. So this is 23. Next is 26. This is 26. And next is 30. So this is our 30. So you can now see from 15 to 30, the residue that we have given, all of them were in that option and all of them fall in a single region of that circle. So you can already imagine that this particular region, all are clustered together, all the residues are clustered together. None of the residues are missing in that option, what was mentioned in the option. All of them are mentioned and all of them are sticking together. There are no other residues that are in between these options, right? So this particular residues are sticking together. So this part can probably be the hydrophobic one because this is the only option that talks about a particular section of the circle. All other options were randomly scattered, but this particular option was only sticking to a certain part of the circle. So our correct option for this particular question, that which of the following residues are likely to be hydrophobic, should be option D. Okay, so the question can come in a different manner as well. Instead of hydrophobic, it could have been hydrophilic as well. That is just they are trying to see whether you do understand the helical wheel and can identify which residues are taking one part of the circle. If you can identify, then you can very easily tell the options. Okay, but whether it will be, here in the question it is written as probably hydrophobic, but it could have been hydrophilic as well. That will depend on the residues. But for now we know that only the last options, those residues are one part of the staying at one part of the circle. So they will stick together and show a single property. Either hydrophobic or hydrophilic, whatever it may be, they will show a single property. I hope this question is clear to all of you. If not, you can tell me. I will discuss again later. Okay. With this, let us move to the next question. Okay, so I will not spend much time in all of those questions because we have to discuss week 3 as well. This is a very easy question, so I'll quickly discuss it. So it states which of the label bonds show free rotation along the peptide backbone. We all know this. The correct answer should be 2 and 5. Okay, that is basically your phi and psi angles bonds, basically NC alpha and C alpha C. We all know that the omega bond, that is CONH bond, that has a double bond character there. Because of its double bond character, it cannot freely rotate. So the bonds that can freely rotate are the bonds around psi and phi. So basically NC alpha and C alpha C. So our correct option should have been option A, 2 and 5. I think all of you know that, so I am moving on from these questions. This is again a very easy question. I will just directly show you the answer. So it states psi, phi and omega are known as, there are blanks that you need to fill, and their rotation angle spans from this to this. There are also blanks that you need to fill. So we all know that psi, phi and omega, these are what angles? You can see Ramachandran angles and another one is dihedral angles. So this is just a nomenclature question. The angles are called dihedral angles. The angles between different planes, they are called dihedral angles. There is nothing, no terms such as Ramachandran angles. We call them Ramachandran plot, from which we get the psi and phi angles, but they are not called Ramachandran angles. They are called dihedral angles, because this is not a thing, the because these angles like dihedral angles, that is basically the angles between two planes, this is not only related to or confined to Ramachandran plots. This is actually widely used in every kind of organic structure. So that is not only limited to Ramachandran plots. So we do not call them Ramachandran angles, we call them dihedral angles. And again we know that in case of psi, phi and omega, so psi and phi angles can be between minus 180 to plus 180. And in the last class we also discussed that the CONH angle can be either 0 or 180. If it is then 0 degree, and if it's trans, then it's 180. So basically our regions goes from minus 80, 0 to plus 80. So the range from all these angles is basically minus 80 to 180. So our correct option should be option B, where these angles are called dihedral angles and the range varies from minus 80 to plus 180 degree. Minus 180 to plus 180 degree. Okay. With this, let us move on to the next one. I want all of you to solve this particular question. Okay, this is a very interesting one. I will not show the answer immediately. So it basically states, okay, so it basically states which is the correct nomenclature for the tripeptide whose structure is shown. So this is a tripeptide that is shown, which will be the correct nomenclature for this. I would request all of you to answer this question because this is a very unique or say good question. Okay, and a easy one as well. So once you have determined it, please do write your answers in the chat box and then I will discuss it. Okay, I will give you some time to figure it out and then write it down in the chat box. Whoever is able to solve this particular question, please write your reasoning as well. Try it out, try your answer. It can be wrong, but that is fine. We are here to discuss. Any of you. Any of you identified it? You can take help of the structure of amino acids as well. If you do not currently remember all the structures, then you can take help from book or internet and figure it out as well. But I really do want at least one or two people to get the correct answer of this question, because this is very basic but very important. Okay, I do have one answer and thank you very much. So yes, the correct answer is D. The structure that is written in here as a tripeptide is not represented correctly. The question is why so? Because each of you know, so whenever in the very first class I told all of you that whenever you see this tripeptide sequence and or any kind of peptide sequence, and to identify the amino acids you are basically needs to break the structure in the position of the amide bonds, in the position of the CONH bond. That is all you need to do, and then you will get the uh, amino acid residues. But one thing that is very important, the conventional thing that we always use, that a peptide sequence will start from N terminal and it will ends at C terminal. Now, apparently it can seem to all of you that what is wrong with it, with the structure, because here is a N terminal and here is a C terminal. So then what is wrong with the structure? But if you look carefully, this is what is this? The very first structure that you are seeing, what is this? This is basically your lysine, okay? But this is the side chain. This is the R group. So amino acid structure is basically like this. You have NH2 or NH3 plus, whatever you can write. One type is COOH, and here remains your R group. So for lysine, basically for K, this particular thing is your R group. The peptide bonds that are formed between the residues are not linkages formed by the R groups. They are formed by the basic backbone, that is this particular backbone which is fixed for all the residues NH2 CH2 COOH. This particular backbone is fixed for all the residues and they are generally involved in the formation of peptide bonds, not the these R groups or side chain groups. That's why they are called side chain groups, because they are not part of the primary backbone of the peptides. So if K is the first one in this case, and so that means it has to be the N terminal. So that means the N portion of K needs to be free. We should not look at the R groups for determining the N terminal or C terminal. N terminal means the basic backbone of the amino acids, that N or NH2 group should be free. So this particular group, which has reacted here with other amino acid, should have been free for the proper writing of peptide. The R group should have been here. This NH2 group should have been free for acting as an N terminal, and the next peptide bond would have been formed with the COOH group of K followed by the whichever amino acid residue is there, that's NH2. Backbones NH2 will come here. Another amino acids backbones NH2 will come here and form the amide bond. So this is where the structure is totally wrong, because wherever you see here or here, the N terminal or C terminal is written in terms of the side chain, but that is not the case. Okay, so be very careful when you see it or when you understand it. The R groups are side chains. They are not part of the primary backbone. The only basic NH2CH2C COOH, they are the ones that form the amide bonds and give you the primary backbone of any peptide sequence, and R groups hang from that primary uh, peptide sequence. Okay, so this was the concept of this question. Hope it is clear to all of you. Now let us move to the next one. So again, this is a easy one, so I would not spend much time in it. It states, which of the following structure is the principal form of glutamic acid at its isoelectric point? So basically, at isoelectric point, glutamic acid should remain as a neutral one. That is the whole point of uh, any, what I should say, the basic thing of isoelectric point, that it should be neutralized basically. So if we look, the correct option for that is option B. This structure should be the isoelectric, this should be the proper structure for glutamic acid at the isoelectric point. And if you look at different pK values of these three groups and the pi value of glutamic acid, you will understand that at that particular pi, the pH value, which is the pi, the NH3 group NH2 group remains as NH3 plus, this one remains as minus, and the side group remains as COOH. So this is the structure for glutamic acid at its pi. So this was a basic or easy one, so I will be skipping this one. Okay, so this is your next question. It states, from the diagram provided, find the correct statements. Okay, so this is your diagram that is shown here. This is your diagram. So these are your option. So 1, 2, 3, 4, there are 4 statements that are written. First statement is A. One second, let me change it again. So this is your A. This is your B part, this is your C part. Okay, so the first statement says A represents an alpha helix at the C terminus. 2 says B represents a beta turn between anti-parallel strands. Oh, I already have an answer, thank you. Okay, 3 statement 3 states C represents an adjacent parallel beta strand, and 4 states A represents an alpha helix at the N terminus. So these were your 4 options. I have 2 answers that I got, but okay, you the same person changed your answer. Okay, then fine. So I already have the answer. So thank you for answering. Let us see whether it was correct or not. Yes, the answer, the changed answer actually that you gave is correct. So let us look. The first option states A represents an alpha helix at the C terminus. A is a alpha helix, that is very obvious from its structure. But if for any kind of protein representation, we know it goes from N terminal to C terminal. So basically, the left hand side should be N terminal and the right hand side should be C terminus. So alpha helix at C terminal, this statement is wrong. It is staying at N terminal. Again, let us look at the second option. So our first option is incorrect. Let us look at the second option. It states B represents a beta turn between anti-parallel strands. Let us look at B and let us look at folding. One arrow is this side, another arrow is opposite. So that means it is definitely an anti-parallel strand. So our second option is correct. Let us look at the third statement. It states C represents an adjacent parallel beta strand. C is, they are stating it is an adjacent parallel beta strand. Whether it is, if you look at it, whether it is parallel, no. The direction of the arrows are opposite. So, it is anti-parallel, not parallel. So, again, this option is wrong. And the fourth option, A represents an alpha helix at the N terminus. We just discussed it at the beginning of this question. So, again, this one was correct. Alpha helix is at the N terminus. So our statement 2 and 4 are correct. So our correct option should be B. Moving to the next one. This is a very easy one. I quickly tell it. So it states, given are the structure of two kinds of beta strands and the hydrogen bonding between the strands. Okay. So identify the correct correlation between the strands and the arrows. So two types of beta stranded structures are given in each of those options and corresponding arrows are given in all of them. You have to correlate that which of the structure correctly represents the arrows, means the structure and the arrows should correlate, which is the correct representation. That is all you need to find. So I will quickly tell you, I think you all know the answer. So the correct option should be C, because why? We already know, if we look at the first structure that is same for all of them. If we see here, all the hydrogen bond, first of all, they are all lateral hydrogen bonds, right? In for the first structure that is given here, in all the cases they are lateral hydrogen bonds. That is the first point. Again, that in the previous class we discussed that if this particular amino acid is reacting with the this NH is reacting with the CO of the second amino acid, again this NH is reacting with the CO of the first amino acid. So basically they are interacting among each other. If we mark them, if this is number 2 residue and this is also say 1 2 3 residue, then in both the CONH bonds that are here, 2 and 3 number amino acids are involved in both the hydrogen bonds, that we have discussed in details in the last session as well. But in case of the second one, first of all, these bonds are not lateral and different amino acids are involved. Like for this particular amino acid, it is reacting with CO with this residue, and again its CO is reacting with another amino acid residue. So it's not like they are interacting with each other. So we just discussed in last session that lateral hydrogen bonds and same interaction between two residues means this is an antiparallel one, and antiparallel beta strands are always represented as arrows of two different direction. That different direction mean at 180 direction, 180 degree opposite direction. But in case of parallel beta strand, the hydrogen bonds are not lateral and they are represented by two arrows that are parallel to each other and going into the same direction. That direction tells us whether it is parallel or antiparallel. So this was the concept you all of you know. So let us look at the next one. Okay, this is a very interesting question again. So I will ask all of you to answer this one. Okay, so it states at pH 7, converting a glutamic acid to gamma carboxyglutamate will have what effect on the overall charge of the protein containing it? So a protein contain, suppose a protein initially contained glutamic acid at pH 7. Now you have somehow converted it to gamma carboxyglutamate. So what will be the effect on the protein's charge? If you modify glutamic acid to gamma carboxyglutamate, what will be the effect of it on the overall charge of the protein? Okay. So there are 5 options. Choose what is the correct one. Okay. And tell the answer in the chat box as well. Okay. So I have one answer and thank you. Yes, the answer is correct. It will become more negative. So basically what you are doing, you just need to know what is the structural change that is happening after you convert glutamic acid to gamma carboxyglutamate. So if you do look at the structures, Suppose it is NH3 plus and this is your COO minus. So here is your structure CH2 CH2 COOH. So this is your glutamic acid. Now you are converting it to gamma carboxy glutamate. What will be the structure? So this is your alpha carbon, this is your C alpha. This is your beta carbon, this is your gamma carbon. So from the name it is very obvious you are just transforming it to gamma position, another carboxyl group you are just adding. So it will be like this, gamma carboxy glutamate. So you are just basically have added another carboxyl group, another acidic group you have added to the protein by converting into gamma carboxy glutamate, and obviously at pH 7 because the pK values of COOH are generally like very less in the region like 2 3 or something. So at pH 7 all of these COOH groups will remain as a decarboxylated form, that is COO minus. So overall the charge of the protein will increase because previously you had two COO minus group and now after converting into carboxyglutamate, gamma carboxyglutamate, you have 3 COO minus group. So it will generally become more negative. Moving on to the next one. Okay, this is a very easy one. I just quickly show the answer. So it states, on a standard Ramachandran plot, where would the beta sheet be located? So you all know this is the Ramachandran plot where the beta sheet regions are mainly found on the top left corner. So that was the basic question. So there are only three things like beta sheets, right-handed alpha helix stated here, and the rare form of left-handed alpha helix, sometimes they are they can be found around the angles of this. Okay, so I will not spend much time in it. Okay, the amino acid sequence LIMF in a membrane protein is expected to be present in or at what position? So this is your peptide sequence LIMF. So what do you expect them to reside in case of a membrane protein? So the important thing is you know your sequence and you know it is interacting with the membrane protein. So where will they go? Will they go at the hydrophobic core, hydrophilic exterior, hydrophilic core, or a hydrophobic exterior? Where will they go if they are interacting with the membrane protein? Okay, so I already have an answer. Others also please try. Okay, so our if I am not getting any other answer, let me show you the correct answer. So one second. Okay, so basically it is stating the correct answer should be hydrophobic exterior. Why so? I think the answer that you gave the correct answer should be hydrophobic core. So the one thing you did not take into account that this particular amino acid sequence is reacting with a membrane protein. So basically what different thing happened here? So first of all, let us look at the residues. It is leucine, isoleucine, methionine and phenylalanine. All of them are hydrophobic residues basically. They all are hydrophobic. That is fine. So the moment you understand that they are hydrophobic, the basic idea comes to that point. So they should go inside a hydrophobic core. That is the idea that you get. But the thing that is mentioned in case of membrane protein. Now what is a membrane protein? Just we learned in the first or second session I guess that membrane proteins look something like this. This is basically membrane proteins has to interact with the membranes of cells, and cell membranes are basically lipid bilayers. So in case of lipid bilayers, this is your hydrophobic region where the tails remain, and you have your hydrophilic head groups. So this is your lipid bilayer. Now if the amino acid sequence, if it has to interact with the membrane protein, then if it has, because this entire part is hydrophobic, if the amino acid sequence that is coming from outside, if it has to interact and have hydrophilic surface, it can never enter through the membrane protein. It can never enter the cell. But to interact with the membrane protein, it needs to have the hydrophobic ones on the surface, so that this protein can go and interact with this hydrophobic region of the lipid bilayer. That is the only difference. Whenever it comes to membrane protein, the scenario is changed. If it was a case of a normal globular protein where it is staying in our physical body, so the hydrophobic residues are staying kind of inside and the hydrophilic ones are outside, that was fine. But as soon as the term membrane protein comes, the scenario changes. You have your hydrophobic residues outside. That is, the correct option should be hydrophobic exterior. Hope this is clear. Moving to the next one. It states a regular right-handed alpha helix comprises 36 amino acid residues. We all know that this is the basic definition for alpha helixes. What is the number of complete turns in the helix? Okay. So I want all of you to answer that. I think you know, you should know. The answer is already found in the name of the alpha helix. Regular alpha helix comprises 36 amino acid residues. Then what is the number of complete turns in case of alpha helix? Just try to figure out what is the other name for alpha helix in terms of number. We have different helixes like 310, like that. So what is the name for alpha helix on the basis of numbers? Just try to figure out that and you can answer it. It is already in its name. Okay, so yes, correct answer is 10. So this is very easy. Let me go back. Which one? Yes. So basically what is alpha helix? What do we write? Alpha helix is nothing but 3.610. We write it. We can also write alpha helix like this, 3.6 by 10. Not by, like it is written something like that. It basically means whenever, and there are other helixes like 310, etc., etc. So basically whichever number is at the top, it means 3 points. If in case of regular alpha helix, 3.6 means there are 3.6 number of residues per turns, and this number at the lower region, it means that how many turns are there. So that means there are 10 turns, and in each turn if you have 3.6 number of amino acid residues. So whenever there are 10 turns and each have 3.6, so overall the number of amino acid will be 36. 3.6 into 10. This is the basic name. So in case of 310 helix also, if the question instead of regular alpha helix, if it is the question for 310 helices, then again it has 10 turns where in each turn you have 3 amino acid residues, That means the overall helix will consist of 3 into 10, that is 30 amino acids. Okay. Okay, I hope it is clear. Moving to next one. This is a very easy one. I'll just tell it straightly. So it states, suppose a tripeptide sequence is written as TIP. Which among the following statement is correct? So you already know that any peptide sequence we see, it starts from N terminal and in case of C terminal. So our correct statement should be C, where it states that proline is in at the C terminus. Yes, proline is it at the C terminus, and threonine is at the N terminus. Yes, threonine is at the N terminus, and this option is correct. You all will be able to answer that. So I will just show you the next question. This is also a very easy question. I will show you the answer immediately. It states in the image below, choose the correct option that represent A, B, C structure of proteins respectively. So these are the three structure for proteins that are shown here. What kind of structures are they? That is the question. So what kind of structure should they be? If you see, yes, the correct answer is what you said is B. Why? Because see, the structure A is basically an alpha helix. What kind of structure is an alpha helix? It is a secondary structure. The next one we can see this different number of alpha helices are joined by loops or turns basically. So this kind of structure, what we call them, whenever these secondary structures are joined among each other by loops and turns, they are called tertiary structure. And the third one is basically the joining or the accumulation of different subunits. Different tertiary subunits are coming together to form a quaternary structure in case of C. So our correct option was option B. So let's see. This is also I discussed in the last class. I think this is the last question for week 2, I guess. So it states anti-parallel beta strands are more stable than parallel beta strands. I think I discussed in details about these questions, that because they have a stronger hydrogen bonds. First of all, they have more number of hydrogen bonds. In case of anti-parallel beta strand, you can refer back to the last week's session video where I showed the structure. So anti-parallel beta strands basically have more number of hydrogen bonds, and because they are lateral hydrogen bonds, so they are basically more stronger hydrogen bonds. So that is the basic thing why anti-parallel beta strands are more stable compared to its parallel counterpart. Okay. Okay, yes, as I thought. So this was the last question from week 2. Now we can directly go to week 3. Okay. So let us start with week 3 questions. We'll continue to 7:10 and then I will give all of you a few minutes break. Okay. So we start with our week 3 session questions. Okay. So this is our first question. It states, sickle cell anemia is one of a group of inherited disorders known as sickle cell disease. You already know that. It basically a disorder that occurs during hemoglobin synthesis due to the difference in the folding of the protein. Okay. So identify the correct answer which describe the mutation. What is the mutation that happens during the folding of protein of hemoglobin synthesis that sickle cell anemia happens? A similar question we discussed in the last session as well, I remember. So from that, try to identify your correct option. What is the mutation that propagates the formation of this disease. Okay, thank you. Yes, the correct answer is option D. That is, mutation happens at the sixth position of the beta chain. That is, glutamic acid is replaced by valine. A hydrophilic residue, glutamic acid, is basically replaced by a hydrophobic amino acid residue, valine. Yes. So I think you all know that already. Let's see the second question. Okay. It states, determining the precise arrangements of atoms within a large protein is possible only through the use of various microscopic and spectroscopic technique. Which is or are the methods mentioned below are used for the structural determination of protein. So there are four microscopy and spectroscopic methods that are written: electronic microscopy, NMR spectroscopy, magnetic resonance imaging, and X-ray crystallography. These four techniques are given. Out of them, which can be used to get the structural determination of a protein? I have one answer already. Yes. So yes, absolutely. The correct answer is option B. So we had four options. Electronic microscopy would no way help you to actually determine its structure, because it's microscopy. You cannot just see the folding of protein by your eyes. Okay. So basically that won't give you any idea. Electronic microscopy, you can just overall see, might see the structure if protein forms some kind of aggregation or some kind of other structures, you can see that. But exact structural determination would never be possible by seeing it, right? So electron microscopy is not the option. The second option is NMR spectroscopy. It is a bit difficult, but it is possible. Many people works in that field. So by NMR, 2D NMR and everything, you can actually determine the structures of protein. The third option is, so first option was incorrect. Second option was correct. MRI, this is nothing going to help, because your protein is, first of all, there is nothing magnetic, first of all, in your protein sequences. So what are you going to see through MRI imaging? What structural idea would you have from MRI? If it does not have any magnetic property, what will you see? So again, this option is incorrect. And the fourth option and the most appropriate option for determining any kind of structure for proteins is X-ray crystallography. You just form a crystal of proteins, and then you take it to XRD and solve it, and know which are the places where the N C alpha C oxygen will be placed from X-ray study. So these are the two options for it. So our correct option was 2 and 4. Moving to the next one. Okay. The question is very long. This is, I would not spend much time. This is just directly taken from the lecture material. So basically it states that if a polypeptide has 100 residues, and which will have 99 peptide bonds, obviously, because if you have 100 residues, 2 residues can form one peptide bond. So obviously 100 residues will form 99 peptide bonds, and therefore they will have 198 different phi and psi bond angles. Each amino acid residue is going to have each peptide bonds. Around each peptide bonds there will be like two angles, one phi and one psi. So overall there will be 198 different angles. Each of these bond angles can be take different stable conformation. But out of this also, what we have, suppose there are this many number of different conformations that are possible. 3 stable conformation, they say that there are 3 stable conformation that can take, and there are like 198 different psi and bond angles that can be happen. So basically total number of conformations that we can achieve is 3 to the power 198. So obviously if a protein wants to folds to its native form, then the protein has to sample through or go through all these different conformation, that is 3 to the power 198, that many conformation, to see that which particular conformation is giving the protein the lowest uh, or the minimal energy value, that is it's going to scan through and then it's going to fold in that particular form. So obviously if protein follows that thing, that okay, I have so many different conformation possible and I would go through each of them and then I will select my native conformation, then you can obviously know that how much longer the time could be taken for one protein folding. But that is not the case. In not the case. If that was the case, the entire biological system would have been failed. So that is not the case. That is the paradox that scientists are trying to solve for years and years and years, that how the protein is able to identify its native structure so quickly. So basically in this question it was asked that this kind of paradox, that it should have been taken a million of years to find a native conformation, yet it has been done by in the mother nature so quickly. So what kind of paradox it is, or what kind of experiment it is called? So you all know this is basically the last option, Leavenworth paradox, that yet to be solved, that how it is uh, the protein is so quickly folding it to its native forms. So this was just a easy question. It was a big question, like to confuse you all, but it was a very easy one. So basically it is your Leavenworth's paradox. Moving to the next one. Okay. Okay, again, this is a big question, but is a very easy one. So it states a homopolymer has only one type of building block called a monomer. Okay, a heteropolymer has more than one type of monomer. That is the basic thing. Suppose, oh, suppose we have these building blocks, and if they are repeatedly attached among each other, then this is called a homopolymer. And suppose if there are different type of monomers and they are attached among each other, then they are called heteropolymers. Basically proteins are heteropolymers because you have different amino acids that are joined. So while a nucleic acid like DNA or RNA is made of only 4 type of nucleotide monomers, you already know that ATGC or ATGU in case of that, AGCU, sorry. So naturally occurring protein in the living sets of system are made of how many types of monomers? This was a very easy question. Yes, the answer is correct. We all know that in case of naturally occurring proteins, we say there are 20 essential amino acids that are important in case of naturally occurring systems. That's why they are called 20 essential amino acids. There are many other amino acids, but they are not non-essential. Only 20 are there. They are the backbone of any naturally occurring proteins in living system. Okay, so this was your question. So after discussing this, I will give you all a break. So let us see. For a protein with 100 amino acids, how many possible sequence will be there? So your protein has 100 amino acid residues. So how many different conformation is possible? Okay. So that is they are asking. 100 amino acid residues and how many possible sequence you can prepare with them. This is your question. I already have one answer. This is just a simple math. Like you have 20 essential amino acid sequence, 20 essential amino acid residues that is going to form the proteins in living system. So out of 20, you have to arrange them in a 100 amino acid sequence. So those 100 amino acids will be taken from this 20 essential ones. So basically, how many possible ways will be there? 20 to the power 100 possible ways. That's how you can arrange them. Any of the 20 can be taken to form this 100 amino acid sequence. It was a very easy one. Okay. So let us start with the sixth question. So it basically states protein S will fold into its native conformation only when protein Q is also present in the solution. Okay. However, protein Q can fold into its native conformation without protein S. So, protein Q may therefore function as a dash or blanks for protein S. So, basically it is stating, once again I have already shown you the answer, so anyway. So, basically it's stating, the question was stating, you have your protein S and you have your protein Q. If S needs to fold in its native conformation, say suppose it has this native conformation, it will need the help of Q to get into its native conformation. Say I am writing N as the native one. Okay. But Q can individually form a native conformation. Okay. So what is protein Q for it? See, if we look at the option, first option is substrate. So no, if Q was a substrate for S, so basically then S will react with Q and give something else, like what the enzymes do. The substrate is a term that is generally used for enzymes, like the enzyme interacts with the substrate and gives you a product. Nothing like that is happening in this particular case or in this particular question. It is that a native folding is occurring. So the for first option, it is out of the question even. The next option, that is the correct option, I will come at the last. Next is protein precursor. Okay. So protein precursor are generally basically are inactive proteins. We can call them inactive proteins that you need to have post translational modification to make it an active one. Okay. So they are basically like themselves inactive. You have to have some modification, do some modification on them, post translational modification on them to make it active. This type of thing is not mentioned in the question. No modification of Q or anything is stated in the question. So again, this option is also out of the point. It is not a protein precursor Q. The last one is a cofactor. Cofactor are basically non-protein things. They can be non-protein organic, some organic compounds or some metal ions like that, which have the role to help the enzyme in the catalysis process. They help the enzyme to act as a catalyst basically, but they are non-protein organic compounds or metal ions like that. So again they are non-proteins. So they have no purpose for folding. In the last one it is written that protein Q can itself fold into its native conformation. That means protein Q is also a protein. Protein Q it is already mentioned, but cofactor means it is not a non-protein, it's totally a non-protein thing, either small molecule or some metal ions. So again our fourth option is wrong. The only option left is molecular chaperone. As far as I remember, at the very first class I probably have discussed about the molecular chaperones. So basically these are the proteins, their job is to find the misfolded parts in other proteins. They are kind of like detectives, you can say. They are finding that whatever misfolding has happened in some proteins, they will rectify those parts and help the protein in to fold correctly. Basically, they are preventing disease. That is their role. So those proteins, so it is possible that whenever the S is folding, some kind of misfolding is happening inside it. So the Q is basically finding that, fixing that thing and helping S to fold in its native conformation. So that is why Q is basically acting as a molecular chaperone. Okay. So I hope this one is clear to all of you. Moving to the next one. It states, given are the steps describing the hydrophobic collapse model of protein folding process. Arrange them in the order in which the protein folding occurs. So there are many methods by which we describe the protein folding, or the different models by which we describe the protein folding. One of them are hydrophobic collapse model. So this question state, these are the four things, or these are the four things that happens in case of hydrophobic collapse. What is the order of the happening? Which step will occur first? Which one will happen at the last? Okay. So I already have one answer. Thank you for answering that. Yes, the correct answer is option C. So basically, in case of hydrophobic collapse, the first thing that from the name you can actually think, basically I have the idea that the very first thing that happens is the formation of the hydrophobic core. Once the hydrophobic core forms, so first the thing that is happen is formation of hydrophobic cores. So this is our first one. Once the hydrophobic core has formed, now the rest part of the proteins we try to form some kind of secondary structures like alpha helices or beta sheets. So that is option statement 2. So after 4 will come the step 2. After step 4, after formation of hydrophobic core, it will happen formation of alpha helices and beta sheets. Once those formed, those secondary structures are formed, they needs to be connected in them. That is the basic thing. So once the alpha helices and beta sheets are formed, they are interconnected by loops and turns. So that is our third step. So after 4 and 2, the next step is 3. And the last step is the polar surfaces or the polar residues remains on the surface of the folded protein to give it stability so that it can interact with the solvent molecules. They interact with the polar solvent, that basically we can think of water in our body system. So they are interacting with water and giving it the necessary stability. So this is the correct order for hydrophobic collapse model. Okay. Hoping this is clear. This is your next question. This is a very interesting one. I want all of you to answer this. Okay. So basically, it states albumin is a large protein which circulates freely in human plasma. Another type of protein is aquaporins, act as a channel for water to enter and exit a cell and are located within the lipid bilayer of a cell's plasma membrane. Based on their physiological locations, how would the tertiary structure of these two proteins most likely compare? Okay, so these are your four options. Read the question very carefully, that where are they found, then you will be very easy for all of you to guess, depending on the location that they are found, that what kind of tertiary structure these proteins have. Okay, read the question repeatedly and select the correct option from the four. B and C are the same. Okay, let me see. As a more non-polar core, the polar outer layer and a more outer layer. Okay. Okay. These two are written same. Okay. So just select between the three. Option B and C are mistakenly given as the same options. Okay. So I have one answer only. So yes, as already mentioned, you mentioned in the chat box that option B and C are same. There's a mistake, I think, while writing the answers. So, so we just omit this one. So out of these three, A, B, and D, the correct option is B, as you have written. So basically, it states albumin circulates freely in human plasma. So what is our human plasma? It consists of water. It is a water based system. Human plasma. Basically it is. So if albumin freely can say move in our human plasma or supposedly water, that means it is very much stable in interacting with water molecules. So obviously if it is very stable in water, that means in the outer layer for albumin there should be what? There should be a polar outer layer because water is polar or plasma is polar. So the outer layer of albumin should be polar, consist of polar residues so that it gives us the necessary stability. And if polar residues are outside interacting with the water molecules, then the non-polar ones will go to the core because they do not like the uh, polar water molecules. So the non-polar ones will move to the core, revealing the polar ones at the outer layer. Whereas in case of aquaporin, it is mentioned that they are found in the lipid bilayer and they helps in for a passage of water to enter and exit in the cell. So they exist where? In the lipid bilayer. So they interacts with whom? They interacts with the membrane proteins that we have already discussed in today's case. So whenever a protein has to interact with the membrane protein and stay stable there, that means they are interacting with the lipid bilayer. So the outer surface should comprise of non-polar things because just as we discussed, lipid bilayer means you have a very non-polar surface at the middle. So your protein has to have non-polar residues on the outer layer to properly interact with the membrane proteins, and then it will have some polar residues inside its core through which the water molecules will pass through. Okay. So the outer non-polar layer is basically giving it the stability, necessary stability or necessary interaction to anchor to the lipid bilayer, and the polar core are which are inside the protein, they are helping the water molecules to pass through it. So our correct option for it was option B. Moving to the next one. Okay. It states which of the following statements is or are incorrect about hemoglobin. So there are three statements. First is hemoglobin demonstrates higher oxygen carrying capacity compared to myoglobin. The second one states there is a covalent bonding between the four subunits of hemoglobin. And the last statement is Oxygenation, the loss of the first oxygen molecule from oxygenated hemoglobin. The deoxygenation process is basically the removal or loss of the first oxygen molecule from the oxygenated hemoglobin uh, protein. Okay, so that promotes the dissociation of oxygen from the other subunits. So basically hemoglobin has four. So if you lose one of the oxygen molecule from one subunit, then the other subunits will also promote the dissociation of oxygen. That is the third point. So out of them, which statements is or are incorrect about hemoglobin? Write your option in the chat box and then I will discuss. There are three statements. Out of them, which one you think, or which how many of them you think are incorrect? Okay, since we do not have much time and I still didn't have any answers, so let me show you the correct answer for it. So let us look at the first one. The first statement is hemoglobin demonstrate higher oxygen carrying capacity compared to myoglobin. So basically how many oxygen can it carry? It can carry 4 oxygen. For hemoglobin it can carry 4 oxygen. Okay, 4 subunits can carry 4 oxygens. But in case of myoglobin it can carry only 1. So obviously hemoglobin demonstrates higher oxygen carrying capacity compared to myoglobin. Then the next one is there is covalent bond. So the first option is correct. Okay, so the first option is a correct option, correct statement. The next statement is there is covalent bonding between the 4 subunits of hemoglobin. Is that whenever it case of protein foldings, and its hemoglobin is basically a quaternary structure. Quaternary, like we already know the primary, secondary, tertiary and the quaternary structure. So quaternary structures are basically the association between different subunits. Is there any covalent bond involved in it? No. They are just accumulating via some kind of non-covalent interaction. So this statement which states that there is covalent bonding between the 4 subunits of hemoglobin, this is a wrong or incorrect statement. Let us look at the third one. It states deoxygenation, that is if you lose oxygen from one of the subunit, it will generally promote the dissociation of oxygen from the other subunits as well. This is what is called the cooperative effect in case of hemoglobin. That is what happens in case of hemoglobin. That is, if you remove one, the other oxygen molecules will also try to get out from the other subunits. That is the basic cooperative effect, the famous cooperative effect that happens in case of hemoglobin. So this third statement is also true. So our only incorrect statement about hemoglobin was statement 2. So for this question our correct option was option A. Okay. Moving to the next one. Which of the following energy terms contributes to the non-covalent component of energy in a protein structure? So there are 4 options. Which of them are non-covalent component of energy in a protein structure? This is a very straightforward question from your literature itself. It is non-covalent interaction basically says yes, your selected option is correct. It is Coulomb energy. So as you have already learned in the lecture of Professor Dasgupta, that is, there are two types of interaction. One is covalent, another one is non-covalent. Covalent interaction means the interactions of the things that are happening in the bonds, bond formation. And in case of bonds, we can compare it to a spring. Two balls are attached to a spring. We can consider a bond like that. So whenever we consider a bond like that, it will have a stretching, it will have a bending, and it will have a torsional energy as well. So basically those all things goes for covalent bonds, covalent interactions. There is another type of interaction in case of proteins, that is non-covalent one, and that is they are like uh, electrostatic interactions, hydrogen bondings, van der Waals interactions, etc. So whenever you are, suppose you are thinking about electrostatic interaction, suppose interaction between NH3 plus and COO minus, this is electrostatic interaction, interaction between two charges, and there comes the law of Coulomb, that is basically Coulomb's energy. So only our fourth option is for non-covalent interactions. All the other three, the first three, are for covalent component of energy for protein structure. Okay. Moving to the next one. Yes, this is another one. It states, which of the following statement is correct? There are four statements. You have to choose the correct one. First, let us see if any one of you can answer this, and then obviously I will discuss this. You can read the options and select the correct one. Okay. So I see you have selected the correct one. Okay, so the correct option is B, but let us look at all the options one by one. The first one states alpha keratin is a globular protein and collagen is a fibrous protein. Is it true? Obviously not, because alpha keratins, they are where it is found. It is basically the components that you found in your hair and nails, that is keratins, made of alpha helices. So it is very obvious. So globular proteins are known because they are very much stable in case of water, whereas fibrous proteins are something that are insoluble in water, basically, because your hair and nails does not dissolve in water, right? So alpha keratin is not that if what it is found in your hair, if it is a globular protein, then it would have been gone every time you take a bath, but it is not. So alpha keratin is a fibrous protein that is not soluble uh, in the water. And again, collagen you can generally find in your skin, bones, any kind of connective tissues where you need the strength. Because if you look at the structures of the fibrous protein, you can see they are look like some kind of ribbons uh, binded together. So basically they give you the strength. They are very strong kind of things. So collagens that are found in our skin, bones and connective tissues, they are also fibrous proteins. Fibrous proteins are generally structural proteins that gives you very strong structures and kind of backup in any kind of thing. They are very you can put strength on them like that. Okay, so both alpha keratin and collagen are the example of fibrous protein. Moving to the next one. Generally, beta sheet structure have repeat sequence of two amino acids, and which are they? So beta sheets regularly have two kind of amino acids. Which are the amino acids in the following options? This is also straightly taken from the lecture material. Which are the amino acids that you frequently find in case of beta sheets? Okay, if I do not see anything, as I have to show many more questions. So correct option for this question was option A, alanine and glycine. If you again go through the lecture materials of this week 3 lecture materials, if you go through it, you can clearly see where the professor has already showed that alanine and glycine, because of their very high flexibility, they are very small, like compared to the others, they are very small, their side chain is very small. So basically they very easily feed in the gaps, because beta sheet, like you are like stacking, you are stacking like different papers one upon another, and in that gap they can easily fit. That's why it is very frequent that you find alanine and glycine in case of beta sheet structures. Okay. Okay, so that is what is written here. Because they are small, they are flexible, so they can be present inside that plane and form the pleated sheets like structures. So basically that's what I said. Moving to the next question, it states the hydrophobicity scale gives values that provide information regarding the relative hydrophobicity of amino acid residues. You already know that is called the Kite-Doolittle scale. So according to Kite-Doolittle scale, alanine has a hydrophobicity index of plus 1.8 and serine has minus 0.8. So what we can deduce from these values of Kite-Doolittle scale? I will not again discuss much about this, because you already know that in case of Kite-Doolittle scale, if you have a positive value, that means what? At least one answer if I can see in the chat box. I am not seeing it yet. So alanine has plus 1.8. Yes. So positive value means it is hydrophobic, and a negative value means it is hydrophobic. Philic. So basically alanine is plus 1.8, so alanine is hydrophobic, and serine is minus 0.8, so it will be hydrophilic. So that is your basic Kite-Doolittle scale. Okay. Moving to the next one. Okay. Yes, that is a very good question. So it states compound A is used as an alkylating agent. It usually reacts with cysteine residues of proteins to prevent them from forming sulfide bonds. Despite being able to react and alkylate proteins, it cannot denature a protein structure. What is compound A out of these four? So be very careful while answering this. It is saying that A is something which will react with the SH groups basically. SH, if it reacts with the cysteine residues, that is the SH groups, and alkylate them. So what will happen? They will no longer form the disulfide bond. To form a disulfide bond, you basically need two SH bonds to come close to each other. If two of them come close, they will form the disulfide bond in the proteins. Basically, this is your reaction. Now, if you alkylate them in place of H, if you place something else, that is what it means if you alkylate. If you do that, they can no longer form the disulfide bonds. A can do that. A can prevent the disulfide bonds formation. But yet it cannot denature the protein structure. So what is it? It is the question. Out of these four options, which compound has the ability to prevent the formation of disulfide bond but yet does not denature the protein structure? Okay, since we do not have much time left, let me discuss this question. So let us look at the options. First option is sodium dodecyl sulfate or SDS. Basically, it's a surfactant. It's a surfactant molecule which has the ability to denature a protein molecules by basically breaking the electrostatic interaction, because itself it is a negatively charged molecule SDS. So basically it can have electrostatic interaction with the protein and it can denature the protein. So our SDS is a well-known denaturant for protein molecules, basically by reacting by electrostatic interaction. The next one is, so it is a denaturant. It can denature protein. So option A will not be, in our case, the correct option. Sodium SDS is not the correct option because it does denature the protein. But our A that is stated in the question does not have the ability to denature a protein. Let's look at the second option. It is stating heating to 90 degrees C. If you heat a protein to such a high temperature, suppose you take the example of egg protein. Okay, if you just simply think about egg, when you heat it to 90 degree, you get the boiled form of egg, that is basically denatured protein. You have denatured the protein. So if you heat a protein to a high extreme condition, then obviously it is going to denature. So again, heating to 90 degree has the ability to denature a protein. Also, simply heating the protein molecule will not magically alkylate the cysteine residues. Okay, it will not magically happen any chemical reaction. So our option B is incorrect again. Let us look at the fourth option first. Urea is a very well-known denaturant for protein molecules by breaking the hydrogen bonds. It basically disrupts the hydrogen bonds and it denatures the protein. And also, urea will generally not go and react with the SH groups. So urea is also wrong. The only option that is left for us, already you all know, the correct option here is iodoacetic acid. So basically, what is iodoacetic acid? Instead of CH3COOH, you place CH3COOI. So what is the advantage of that? CH3COOI. What is the advantage? So basically it makes it very easy to replace or react with the SH groups. So S minus can very easily interact with the carbonyl group of iodoacetic acid. So whenever that happens, the SH groups transform into SCOOCH3, kind of like, okay, SCOCH3. Now if they are becoming alkylated, they can no longer form the SS bond. But it does not necessarily, iodoacetic acid does not necessarily interfere with the other stabilizing factor that are present within the protein, like breaking the electrostatic interaction, no. Breaking the hydrogen bonding, no. It does not interact with the other things. It just simply prevents it to form the disulfide bonds. So it does have the ability to alkylate the proteins and prevent SS bond formation, but it will not necessarily lead to the denaturation of protein. So our correct option in this case was option C. Hope this is clear. Moving to the, I think probably this will be the last question or another one. Okay, so this states egg white protein. I just gave you the example in the last question. Egg white protein, ovalbumin, is the best visible example of protein denaturation. You can see it literally. when heat is applied to it, when the egg is poached in water, we can see the egg white changes its color and texture. So basically the protein is denaturing. So which structure of the ovalbumin remains least affected by this denaturation? So these are the four structures: primary structure, secondary, tertiary and quaternary. So whenever you are boiling the egg, which structure remains least affected? Gets affected obviously, but which of the structure remains least affected by the denaturation process? Yes, obviously the option is correct, that is said is primary structure. Primary structure means basically your peptide bonds, the bonds between the amino acid sequences. You can break the other bonds like that are responsible for protein stable structure, basically hydrogen bonds, electrostatic bond, van der Waals forces and everything. You can break them at extreme condition, but that is not exactly enough to break the peptide bonds as well. As primary structure is basically means the peptide bonds, so they are the least affected ones during the whole denaturation process. I think this is a very easy one. I will just quickly say we have already discussed this. In the next question it states a regular right-handed alpha helix has how many strands? I have just discussed this in a previous question that regular right-handed alpha helix means it is 3.610. We can write it like that. And the 3.6 means there are 3.6 amino acid residues per turn. So our correct option for this question is option D. There are 3.6 amino acid residues per turn. Okay. This is again a very easy question. It states peptidases or proteases are enzymes that catalyze peptide bond hydrolysis. They cleave a bond between which of the atoms? This is a very easy question because from the name you can understand peptidases means it breaks the peptide bond. That's how the enzymes are named generally. In their name you can very easily understand what is their role. So peptidases, that means they have the one role, break the peptide bonds. So where are the peptide bonds you can find? Which of the atoms they form peptide bonds? In and carbon, not the C alpha. C alpha has that side chain group. So that does not form the peptide bond. C and O, oxygen is upward, CO. The main bond is between nitrogen is carbon. So the second option is not correct. Again in the option C it states C alpha. C alpha is no way involved in the peptide bond formation. So which atoms form the peptide bond? Nitrogen and carbon. So they are basically the one that will be targeted by peptidases and proteases. I think we can take another one. This is the question we just asked a few way back, so I'm gonna skip this. Okay, so I think 19 and 20 question, up to question 20 we will solve and then I will end today's session. So question number 19 states myoglobin is a globular protein found in our blood. It acts as the oxygen carrier in muscles. We all know that. If we consider that a myoglobin moiety contains a lysine and a leucine residue, where are they likely to be present in the protein? So again, the very answer of this question is written in the question. The very first point that you need to notice, myoglobin is a globular protein and they are found where? In our blood. So, what is the core structure of globular protein? That in the core you have the hydrophobic residues and the outerwards you have the hydrophilic residues. And that is why they will be stable in the blood or plasma because there are hydrophilic residues on the outer layer. So, if this question states as myoglobin has lysine and leucine, out of them Lysine is what? Lysine is a hydrophilic amino acid residue. So where it will go? It will go in the last option, exterior, because it is a globular protein. So lysine molecules will go to the exterior of the protein, globular protein. And leucine is a hydrophobic amino acid residue. So obviously where it will go? To the core or the interior of the globular protein. Moving to the next one. So I think this will be the last question that we can take today. So it states, which of the following statements is not true regarding the comparison of alpha helix structure and beta sheet structure in proteins? So you are comparing alpha helix and beta sheet, and which of the following statement is not true? So the first option states, each may occur in typical globular proteins. Both of them, alpha helix and beta sheet, any of them can occur in case of globular proteins, right? They can, you cannot. You have to look at the sequence. That whether it is alpha helix or forming beta sheets, you have to look at the sequence. But each may appear in case of globular proteins. Is this a wrong statement? No, it is a correct one. Next option is, okay. Next option is, each is an example of secondary structure. So obviously alpha helix and beta sheets are the options of secondary structure. You already, all of us know that. So that option is also correct, no, a correct statement. The third option is, each is stabilized by interchain hydrogen bonds. Now if you think about the structure of alpha helix, if it is a alpha helix and the hydrogen bond remains like this, interchain hydrogen bond means two different chains should be involved. That is why it is called interchain. But in case of alpha helix, the hydrogen bonds exist between its own segments. They are called intrachain hydrogen bonds, not inter. There is no second party involved. But in case of beta sheet, if you look, if these are the beta sheet, then these are the hydrogen bonds supposed to. So there are, this is one chain, this is one beta strand, this is another beta strand, and there is hydrogen bond. So this is inter-hydrogen bond. But in case of alpha helix, it is intra-hydrogen bonds. But in the third statement, it is stated that each is stabilized by interchain hydrogen bonds. That is not true. Alpha helices are stabilized by intrachain hydrogen bonds. So our option C was incorrect statement. And the last option, just let us look at it. Peptide bond in each is planar and trans. Obviously, cis peptide bond is not favored. Trans are the ones that are stable, and peptide bond in each of the cases is planar. Obviously, that has to be because it does have a double bond character. So it try to remain in a plane. It will not try to rotate. It does have a planar structure. So our fourth statement was correct as well. So the only incorrect statement was option C. With this, I will conclude today's session. We have many more questions, important questions left. And like I did in today's session, in the next session also, I will start with those. So up to 20 we have done. So there are up to like 36 questions. Okay. So 16 questions left. 16 or 15 would be there. Some questions are repeated. So we'll solve those questions in next session first, and then we'll move forward to week 4 sessions. And again, I would like to say, if any of you have any doubt, feel free to ask. I will solve it and try to discuss it in the class.